acid and protein synthesis. So, starting with the topic. 1 second. Right. So when we are talking about before we can talk about protein synthesis, right? Before we can talk about how proteins are made, so we have to do protein synthesis, and we have to talk a little bit about DNA replication. So whenever we are talking about these two main things, before we can do that, we actually have to understand what the structure of your DNA is. What is going to be the structure of your DNA? So before we can understand the structure of the DNA, we have to understand the structure of a nucleotide, because nucleotide is the monomer of your DNA or your any nucleic acid. So when you talk about nucleic acids, you can have DNA, you can have RNA, multiple types of nucleic acids are there. So nucleotide is basically your mononucleotide is basically your monomer for your nucleic acids. Now, when I am talking to you about monomer, we need to know in detail what the structure of monomer actually is. So our monomer or our mononucleotide is made up of one sugar component. The sugar component it is made up of, it is DNA, we have deoxyribose, and in RNA, we have ribose sugar. The difference is literally in one oxygen. Deoxyribose has one less oxygen than ribose sugar. So both of them have got 5 numbers of carbon, 5 carbon atom. Both of them will have the same number of carbon atoms. The rest of the structure is going to be pretty much the same. There's going to be no difference in rest of the structure. The only difference is going to come that deoxyribose has one less oxygen compared to your ribose RNA, compared to your ribose sugar. So that is what the first part of a mononucleotide is. We have a sugar component. Attached to the sugar component, you will have a phosphate group, and along with the phosphate group, you will also have a nitrogenous base. So that is the primary structure, a 5 carbon sugar. So a 5 carbon sugar generally is telling you the general name of a 5 carbon sugar is that we call it a pentose sugar. Pent meaning 5. So we call it a pentose sugar. And then inside pentose sugar, we have 2 options. Either it can be deoxyribose or it can be ribose. So since that is a 5 carbon sugar, we call it a pentose sugar. And then it can be further inside pentose. You can have different types of further different sugars. So we have deoxyribose and ribose. We have our inorganic phosphate group. And then we have our organic nitrogenous bases. Generally speaking, in our nitrogenous bases, we have inside DNA, we have 4 major nitrogenous bases. We have adenine, thymine, guanine, and cytosine. When you talk about RNA, RNA does not have thymine in it. In RNA, thymine is replaced by uracil. So that is what the general structure of your, that's what the structure of mononucleotides are. Now, let's discuss how these mononucleotides will come together to actually give you and make your structure of DNA. So, once again, let's take a look. We have our inorganic phosphate group. As you can see, phosphate group has a negative charge on it. So your DNA actually has an overall negative charge, and your DNA is an acidic molecule. So first thing, DNA is an acidic molecule. Second thing, your phosphate group has a negative charge on it, which is why DNA has a negative charge all over it. Second component is our sugar, ribose and deoxyribose. And you can see the difference over here if you look at carbon number 2. So carbon number 2 over here, you can see that the OH, we have an OH group. While on deoxyribose, we only have hydrogen. So that is why we call this deoxyribose. That is literally just one oxygen that is not there on carbon number 2. Then we have our nitrogenous base. Generally, we can divide our nitrogen bases into 2 classes, purine bases and pyrimidine bases. You don't have to go into detail of what is purine and what is pyrimidine. What you do have to remember is which base is a purine and which base is a pyrimidine. So you have to remember which is which, but you don't have to go into details of what they are. Generally, we have purine bases and we have pyrimidine bases, and the differences in the structure. Purine bases generally have 2 rings inside of it. Pyrimidine bases are made of just 1 ring. Okay, so purine bases have got 2 rings, pyrimidine bases have 1 ring. Adenine and guanine are both considered to be purine bases. Thymine, cytosine, and uracil are pyrimidine bases. So you don't have to go into details of what do I mean by purine and pyrimidine. You just have to remember which is which. So adenine and guanine will be purine. Thymine, uracil, and cytosine are pyrimidine. And that is the structure of a mononucleotide. Now, one thing that we have to clear is what is the difference between ATP and adenine, right? We have ATP and we have adenine. There is basically a difference when I'm going to talk about it. Okay.
Yeah.
ATP is adenosine triphosphate. What is adenosine? When you take adenine and you attach it to a sugar. When you take adenine and you attach it to a sugar, that becomes adenosine. So ATP is adenosine triphosphate, adenine attached to ribose sugar. So that is adenosine. When we talk about the nitrogenous base adenine, or when we talk about adenine in the DNA, we are just talking about the nitrogenous base. When I say that adenine is going to bind with thymine, or it is going to make a bond with thymine, I am talking about the nitrogenous base. When I say ATP, I'm talking about adenosine. So adenosine is basically adenine, the same nitrogenous base, but combined with ribose sugar. So that is going to be your adenosine. And then attached to the adenosine, you can have different phosphate groups. If you have just one phosphate group, we call it adenosine monophosphate. If you have 2 phosphate, yeah, Aisha, you want to ask something?
Yeah, miss. So it's always going to attach to ribose sugar. It can't be deoxyribose sugar.
Yeah, it's ribose sugar. ATP is with ribose sugar.
Only ribose?
Yeah, ATP is ribose.
Okay.
Deoxyribose is in DNA. ATP will have ribose sugar in it.
Okay.
And if it has got 1 phosphate group, we call it adenosine monophosphate. If you have 2 phosphate groups, we have adenosine diphosphate, and 3 phosphate groups is adenosine triphosphate, and that is the structure of ATP that we have to talk about. Now, so generally what happens is that whenever you're using ATP for energy, right? Whenever you're breaking down ATP for energy, what do we normally do? We normally, one second. We normally do a hydrolysis reaction and we break down our ATP into ADP. So whenever we are breaking down ATP for energy, whenever we're using ATP for energy, we are breaking it down from ATP to ATP. And generally, when we are making energy, when we're making ATP, and where do you make ATP? You make them in your mitochondria. So when you're making ATP inside the mitochondria, you do the opposite. You take your ADP and you turn it back to ATP. So that is how ATP actually works. Now, let's go over how the structure of DNA is actually going to be made. So DNA structure is made. First of all, we know that DNA is a double-stranded structure. It is a double-stranded helical structure. So you need to have 2 strands of it. First, let's talk about a single strand, and then we will talk about the second one. The single strand of DNA is actually made of multiple nucleotide bases coming together. So multiple mononucleotide will arrange on top of each other to give you your polynucleotide, or to give you a single strand of DNA or RNA. Inside it, the bond, how will they attach to each other? They will attach to each other with a bond that will form between the phosphate group and the ribose sugar. So a bond will form between the phosphate group and the ribose sugar. And this bond is known as your phosphodiester bond. So a phosphodiester bond is going to form between the phosphate group of one nucleic acid or one mononucleotide and the sugar of another mononucleotide. So sugar of one mononucleotide and phosphate group of the next mononucleotide, between them you will end up getting a phosphodiester bond, that is a covalent bond. And after that, when you're going to take a look into it, they will keep on arranging on top of each other. They will keep on arranging on top of each other to give you a polynucleotide. And this entire structure at the back, this structure, all together, if I give it one name, I call it a sugar phosphate backbone. Sugar phosphate backbone. So this is the backbone of the DNA. We call it a sugar phosphate backbone. And what is the sugar phosphate backbone made up of? It is made up of phosphate bond, covalent bond between the phosphate of one mononucleotide and sugar of another mononucleotide, and the bond is phosphodiester bond. So this is how one chain of DNA or RNA will be made. DNA is going to have deoxyribose and RNA will have ribose. So that is how one chain is going to be made. After one chain is complete, now DNA is a double chain structure. RNA is fine, RNA is done. DNA is a double chain structure, and we need to have our second chain as well. So the second chain will arrange itself in such a way that your complementary bases are going to arrange in front of each other. So complementary bases, what are complementary bases? Adenine and thymine are complementary to each other, which means that they will always arrange in front of each other. Cytosine and guanine are always going to be in front of each other, which means that they are complementary to each other. So your mononucleotides will start to arrange in front of complementary bases. So in front of thymine, you will always get adenine. So complementary bases are going to start to come, and between the complementary bases, you will get hydrogen bonding. So the phosphodiester bond was a covalent bond. The bond between your complementary bases is hydrogen bonding. And specifically between thymine and adenine, you have two hydrogen bonds, and between cytosine and guanine, you get 3 hydrogen bonds. So you're going to start getting hydrogen bonds between your complementary base pairs. That is the basic structure of DNA. Now, one thing you're going to notice is that one chain is straight and the other chain is upside down. The term we use for it is antiparallel, because they are parallel to each other, but in the opposite direction, right? They are parallel to each other, but they're parallel to each other in the opposite direction. And basically, you will see two terminologies. You will see 5' and 3'. So 5' and 3' basically tell you the number of carbon in the sugar. Okay. So for example, if I'm going to take a look over here, if I take a look at this particular carbon, this is my phosphodiester bond. Hold on, let me just see if I can find a better picture. Just give me 1 second. I'm just trying to see if I have a better picture of it. Okay, so I don't have, okay, so basically your 3' end and your 5' end basically shows the carbon atom in a deoxyribose to which the phosphate group is bonding. So if you're going to take a look over here, for example, this particular phosphate group is bonding at carbon number 5. Carbon number 1, carbon number 2, carbon number 3, and then you have your carbon number 4, and you have your carbon number 5. The carbon number 5 actually comes like this. If you look at the structure over here, so this is your carbon number 5, right? So if it is binding to carbon number 5, is the phosphate bond is being made with carbon number 5, that particular end, this is just a way of giving terminology to the ends, or just a way of giving a name to the ends, so we can talk about it easier. So this particular end will be known as 5' end, meaning that the phosphate group is with the bond is being made with carbon number 5. If you look over here, this is carbon number 1, this is 2, and this is carbon number 3. So over here, the phosphate, the bond is being made with carbon number 3. So this end is going to be known as the 3' end. So one end is known as the 3' end, and the other end is going to be known as the 5' end. So you don't have to go into details of it. They're not in your syllabus. I just wanted to explain to you that the two chains will be in opposite directions to each other. The terminology we use is anti-parallel. So one chain is going to be going from 5' to 3'. The other chain is going to be going from 3' to 5'. So that is what I mean, that the chains are going to be in opposite directions to each other, and they are going to be, so with the term we use for it is that they're anti-parallel to each other. So you don't have to go into details of it, by the way, the 5' and the 3'. I just wanted to give you a rough idea of what we mean by it, just in case you see it in the exams. But that is not in your syllabus. All you have to understand is that both of them are in opposite directions. There is going to be complementary base pairing, and your two chains are going to arrange next to each other in an anti-parallel direction. Once this entire structure is done, once the two chains have arranged themselves in a complementary way, the DNA is going to turn into a helical structure. The DNA is going to end up turning into a double helical structure. And at every turn, you have 10 base pairs. So at every turn, you will have 10 base pairs. Let me draw this. So over here, between this every turn, you will have 10 base pairs. That means 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So at every turn, you will have 10 nitrogenous bases pairing with each other. And that is going to be the complete structure of DNA. Any confusion so far? Anything you want to ask?
No.
All right. So that is basically how it happens. Now, before we get in, that is what the structure of DNA is. Now we need to go over DNA replication. How does the DNA divide? But before we go over how the DNA divides, let's first go over our theories. What are the theories? And then we will actually look at an experiment to explain those theories. Okay. So there are multiple theories available to explain how the DNA actually replicate. Namely, there are 3 theories. Let's start with the first one, that is conservative replication. This is an older theory that we don't really believe in anymore, right? Conservative replication is a theory that we don't really believe in anymore. It's not, what we are working with. It is an old theory. So the older theory basically stated that you would have your DNA. Your DNA would unwind. It would separate into 2 strands. And after it's separated into 2 strands, you would get a copy of each strand. And what would happen? The old strands would combine, the new strands would combine with each other to make the new DNA. And the old strands would combine with each other to make another DNA. So that was conservative replication, that the old strands will combine with each other and it is going to make one DNA, and the new strands will combine with each other and they will give you a new DNA. So this was conservative replication, in which both of the original strands were being conserved. Both of the original strands were being kept. Both of the original strands were coming back to join with each other. This is conservative replication. Before I explain to you the experiments, let me just tell you all the theory. The second theory we that we nowadays follow and that we now believe is known as semi-conservative replication. Semi-conservative replication states that, you would open up your DNA and you would make a copy of each strand. After you would make a copy of each strand, the one new DNA and one old DNA will combine with each other to give you two new strands. So one old DNA and one new DNA will combine with each other to give you your two new strands. This is semi-conservative because in each DNA strand, only one original strand is being conserved. So one original strand is being conserved over here and one original strand is being conserved over here. So this is known as semi-conservative replication, where one old strand and one new strand is present in the DNA. After that, the third theory, and that is also something we don't really believe in anymore, is known as dispersive replication. I'm not going to details of dispersive replication, but basically dispersive replication said that in each new molecule would be made of old bits and new bits scattered randomly throughout the molecule, which means that this would be your original strand, this would be the copy, and when they would combine with each other, you would get something like this. So you would have small areas of the old strand, small areas of the new strand, and they would all sort of be mixed up with each other. So small bits and pieces of the old strand, small bits and pieces of the new strands, and all of them would come together and they would basically end up giving you your complete DNA. This is dispersive replication. We don't believe in this either. What we have proven, science has proven, is that semi-conservative replication is basically the theory that DNA actually follows. How did we end up doing it? How did we end up proving it? Before we get into that, it is clear, right? What the three theories are?
Yeah, ma'am. Let's do
All
the
right.
questions on the three theories.
Sorry?
Will we be questioned on the theories, like three of them?
Yeah, exactly. You will. They give you MCQs like this, where they will give you the experiment and then they will ask you, or they will say, what result would you expect to see at the end of the experiment? Or they ask you and they say, for which theory, what experiment would you expect? So generally you're not going to get essay questions on the replication. You might end up getting 1 or 2 mark questions on it, but you will see MCQs related to this. So that is why we have to do the experiment part, because you will get MCQs where they'll ask you to like identify like what will be the results of this particular replication. So when we are talking about let's talk about conservative replication. So in conservative replication, what the scientists actually started to do is that they said, why don't we go ahead and we start with, let's go ahead and test. Let's go ahead and do an experiment to see which method of replication is being followed. So what they went ahead and they did is that they took two isotopes of nitrogen, nitrogen 15 and nitrogen 14. Nitrogen 14 is of course light and nitrogen 15 is of course going to be dense. So what they went ahead and they did is that they took the original strand and they made the original strand of nitrogen 15. Then after they had the original strand, what they did was they added, so the nitrogen 15 was just for the original strand, that is it. After that, they added nitrogen 14 into the mixture. So that when the new strand was made, the new strand was made of nitrogen 14. Now what they went ahead and did is that they went ahead and did an experiment where they took the DNA, where they took the resultant DNA and they actually did centrifugation and they separated the DNA. They separated the DNA strands that were being made based on their mass. So they started to separate the DNA that were being made based on their mass. Actually, hold on. So they started to separate the DNA bands based on their mass. The DNA that was being copied, they would separate it based on their mass. So first, let me tell you what result we would expect from conservative replication. What result would you and I expect to get for conservative replication? And then I will tell you the result they actually got. Okay. So generally speaking, in conservative replication, the new strand will combine with each other. So that means both the strands will have a mass of 14 and 14 because we're using nitrogen 14, and that will combine up to 28. The old strand would have a mass of 15. The old DNA strand would have a mass of 15 and 15. And when you add that up, you will end up getting a mass of 15. So when you were putting the DNA inside your test tube and you were letting it separate, if conservative replication was true, you would end up seeing two bands of DNA. You would end up seeing a light band of DNA that was made of 28, and you would end up seeing a heavy band of DNA that would be made of the collective mass of 30. That was made of nitrogen 15. So this is what you would expect to see. But this is not what we actually saw when we did the experiment. What we saw was a different result. So what they saw was that when we first did the replication, when we did the first replication, we only got one band in the middle. Let's see how that happens. So this is our DNA and this is the copy. This is what you would end up expecting at the end of semi-conservative replication, right? That is what you would expect at the end of semi-conservative replication. So if you look over here, this is going to be 15. This is going to be nitrogen 15. And after that, you have nitrogen 14 and nitrogen 14. That means the combined mass of each DNA is going to be 29, 29. So when you do the experiment, you will see a single band it's not going to be heavy, it's not going to be light, it's going to be medium, and it is going to be a single band because both the DNA are going to have the same mass. This was the result of the first replication, not the second. The first replication. The first time you let the replication happen, this is the result you would get. And this is the result that the scientists got. After that, they said, let's do another replication. Let's keep on repeating the process. Let's do a second replication. So they continued from where they left off. They had a DNA, semi-conservative replication. They had a DNA like this. So they said, let's go ahead and let the replication happen again. Let's go ahead and let this DNA keep on replicating. So what happened when they did the second replication? They ended, the DNA will unwind. And after the DNA will unwind, you will again get new copies. When you will end up getting your new copies, so now what's going to end up happening? This copy is going to have nitrogen 15, but the new copies are all going to have nitrogen 14 because the only isotope of nitrogen available is nitrogen 14. So now when your DNA strand will form, you will get something like this. So this strand is going to be nitrogen 15 and the rest of them all are going to be nitrogen 14. So this strand will have 15 plus 14, 29 and 14 plus 14, 28. So this time when you do the replication again, so we are talking about semi-replication. It is just that we are doing the first replication and the second replication. So when you're doing the second replication, you will end up getting one band that will be medium in weight. That will be because of your 29, this one. And then you will end up getting one band that is going to be your 28. So, and then they kept on repeating it and they kept on looking at the band. So this is the result they were getting, and they actually ended up saying that they ended up concluding that, yes, we are actually, if you look into it, semi-conservative replication will explain the bands we are getting. If we talk about conservative replication, we should have gotten two bands, one heavy and one light. But over here, we are first getting a medium band, and then we are getting a medium band and a light band. And so we are getting different results. So then they concluded that semi-conservative replication is the theory that you actually have to follow. So you will get MCQ questions where they're going to give you the tubes and they're going to basically ask you based on that. So that is what you will have to go with. Okay, making sense? Any confusion in this?
No, 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 no.
All right, so that is it. You don't have to go into any more detail than that. Now let's actually talk about how the process of replication takes place. The first thing you have to do is you have to unwind the DNA and you have to open it up into two strands. We have to go ahead and we have to unwind it and we have to open it up into two strands. That is the first thing that we have to actually do. Who does it? Who helps us in opening up the DNA? Who helps us in basically separating the DNA into two strands? It's an enzyme. We call it DNA helicase. So DNA helicase is going to come and it's going to separate your DNA into two strands. Once your DNA has been separated into two strands, what do we have to do? We have to bring our nucleotides, we have to bring our complementary nucleotides and start arranging them in front of the template strand. So this is known as the template strand. The strand that you're making a copy of, that particular strand is going to be known as the template strand. So you're in front of your template strand, you will start getting your complementary bases. Generally speaking, the bases have two phosphate groups attached to them. Okay. So they have an extra phosphate group. The extra phosphate groups activates the nucleotide. It activates the nucleotide so that the nucleotide can then take part in the reaction. After that, what is going to happen? Your activated nucleotides are going to arrange on top of each other depending upon complementary base pairing. So whatever your complementary base pairing is, depending upon that, your activated nucleotides are going to arrange on top of each other. As they're going to arrange on top of each other, the extra phosphate group is going to be released. The extra phosphate head is going to be released, and your phosphodiester bond is going to be made. So the extra phosphate groups are going to be released, and the activated nucleotides are going to make a bond with your sugar. They're going to make your phosphodiester bond. And who is going to help you make this bond? DNA polymerase. DNA polymerase is then, 1 minute. Your DNA polymerase is basically going to be present. Your DNA polymerase is going to make sure that only complementary base pairs can arrange on top of each other. So if the complementary base pair is not there, DNA will not make the DNA polymerase will not make the phosphodiester bond. So DNA polymerase will make sure that complementary base pairs are arranged in front of each other, and then it will make your phosphodiester bond to end up completing your chain. So that is how the process of your entire DNA replication takes place. Just to give you a, your complementary strands will be made, and after your complementary strands are going to be made, the one new strand and one old strand will bind with each other. The hydrogen bonds will be made between the nucleotides, and then the two chains are going to turn into a double helix, and that is basically going to that is basically the reason that you're going to end up getting your replicated DNA. Now, one fact I would like to add, you don't have to do details of DNA replication, but generally I'm telling you that your one chain of the DNA is made in one continuous direction. Okay. So it will start when you're talking about your DNA polymerase travels in a specific direction. So remember, your two chains are in opposite direction. So one chain is going to be made in a continuous manner. It is going to be made in a continuous manner because it is because the DNA polymerase travels in this direction. The other chain, it cannot be replicated directly into one long chain because the DNA polymerase does not travel in this direction. So what happens is that one strand is going to be made in a straight chain. One strand is going to be replicated continuously. The other strand is going to be replicated in small pieces. So one small piece is going to be replicated, and then a small piece, then a small piece, and then they will all join together. So you don't need to go into details of it. There are actually quite a lot of details of it, but you don't have to go into that. What you have to basically understand over here is that one chain is made continuously and the other one is made in small sections and then joined together. And that is because of the fact that they're anti-parallel to each other. So that is why I mentioned that before, that they're anti-parallel to each other. So that is the significance of it. So, but you don't have to get into details of it. I just mentioned the fact because they have given they have mentioned it in questions. So that is why I wanted to mention that small fact. Okay. So any confusion in DNA replication, we all clear related to how DNA replicates?
Yes, yes, it's clear.
Aisha, all clear? Any confusion?
Yeah, it's all clear.
All right. So when now we're done with this, we have to go ahead and talk a little bit about our protein synthesis. Okay. Now there are two parts for protein synthesis. We have transcription and we have translation. Right? Transcription is when you are transcribing or you are copying the DNA and using it to make an messenger RNA. By the way, there are multiple types of RNA. Messenger RNA, ribosomal RNA, multiple different types of RNA that you're actually, if you decide to ever do it on a postgraduate level, you will understand, we'll go over that. So we have messenger RNA, and the another form of RNA is ribosomal RNA. Ribosomal RNA is actually made in the nucleolus. The nucleolus that you have inside the nucleus, the function of the nucleolus is to make ribosomal RNA, and ribosomal RNA is assembled then to make your ribosome. So ribosomes are made up of RNA. So the initial process of transcription is when your DNA is going to be transcribed to give you your messenger RNA. Then your messenger RNA is then going to be read by the ribosomes. The ribosomes will read the messenger RNA, and after reading the messenger RNA, they will end up giving you your protein chain. So that is translation. When messenger RNA is read and a protein chain is made, that is going to be known as translation. So we have to do both the processes. We have to do transcription and we have to do translation as well. So let's go ahead and start with the first part, that is your transcription. How is transcription going to take place? So generally you start by opening up the DNA. You don't have to open up the entire DNA, only the section of DNA that you need. For example, let's say we're talking about the cell of the pancreas, right? So what is the function of cell of pancreas? To make insulin, okay? So only the region of DNA that has insulin in it, that region of DNA is going to unwind. After that region of DNA is going to unwind, what is going to happen? Your RNA is going to be made. So we have two strands. One is known as the template strand, and then we have the other strand. So only the template strand is the one that is going to get transcribed. So in transcription, it's not both the strands. In DNA replication, both the strands were getting copied. Over here, you will only make the complementary strand of the template strand. Only the template strand, one strand will be used, not both of them. So what is going to happen, that the strands are going to unwind, and the template strand is going to be used to make your messenger RNA. Now, what is going to happen? Once again, just like in the process of replication, complementary nucleotides are going to start to pair up with your DNA. So whatever is the nucleotide of DNA, in front of that, you will end up getting your complementary nucleotides. In front of that, you will end up getting your complementary nucleotides. And then an enzyme that we call RNA polymerase, that particular enzyme will come and it will start to make the sugar phosphate backbone. Just like DNA polymerase, it is going to come and it is going to make sure that the base pairs are complementary. It's going to make sure that only the complementary nucleotides are attached, and it is going to complete the sugar phosphate backbone. So an entire chain of your messenger RNA is going to end up forming. Now, when we are talking about our messenger RNA, when I'm talking about the entire process, so generally we have something that is known as your codon. Okay, what is a codon or what is a triplet code? When I'm talking about the DNA, on the DNA, I have multiple nitrogenous bases. Right? I have multiple nitrogenous bases. So, generally speaking, what is going to happen if I combine three of the bases, on the DNA or on the RNA, those three bases will give me something I call the triplet code. Triplet meaning three, and code means that it is going to be coding for something. So when I read the three bases together, they will give me a specific code. And that code is going to be specific for a certain amino acid, right? So we have our triplet code, and the triplet code is going to tell me, when I read the three nucleotide bases together, it will tell me what amino acid should come and attach over it. So that is how the DNA actually works. That is how the DNA passes on the information. So what is going to happen, that your messenger RNA is going to be formed and your RNA polymerase is going to attach the entire process. Once the messenger RNA, for the section of DNA that has your, that has your information for making insulin, once that entire area is going to be transcribed, your messenger RNA is going to detach, it is going to move away, and the messenger RNA will be made. Once the messenger RNA is made, the messenger RNA is modified. You don't have to go into details of modification, but the RNA is modified. How is the RNA basically modified? When you talk about your DNA has two sections. It has coding sections and it has non-coding sections. So there are sections of DNA that actually code for some information. How to make insulin, how to make glucagon, how to make enzymes, how to make this, how to make that. So you have areas of DNA that actually give you information on how to make different things, how to do different processes. But majority of the DNA is actually made of non-coding region, which means that majority of the information on DNA does not really code for anything. It does not really, it's not for proteins, it's not for enzymes, it's not for anything. So large areas of DNA are non-coding. They actually carry no information at all. So the scientists to this day, they're still working on the function of non-coding DNA. Why do we have such a big structure of DNA that does not give us any information or that does not code for anything? We don't know. They don't know. They're still working on it. They haven't managed to figure it out. But when your messenger RNA is copied, it has both areas. It has coding areas and non-coding areas. Everything is going to get transcribed. Once the messenger RNA is made, it will be further modified. So what you will do is you will cut out the non-coding regions. Whatever non-coding regions are there, they will be cut out. So the messenger RNA that will be left behind will only have the coding regions of the DNA. So after that, once your messenger RNA has been modified, it is complete. It will leave the nucleus to the nuclear pore and it will enter into the cytoplasm. Any confusion in this?
Please, can you repeat that once again? The coding one?
All clear, Aisha? I hope no confusion.
Yes, I was saying, can you repeat the last part, the coding one?
Yeah. So when you talk about your DNA, there are two regions of DNA. You have coding region and non-coding region. The coding region of the DNA actually carries the information for different processes inside the body, or for making different, what do you call it, different proteins, or making different parts of the body. Then you have non-coding region. Non-coding region does not seem to carry any information at all, or it does not seem to carry information uh, related to anything. It just seems to, it just seems that we have these huge sections of DNA that are not used for anything, like huge parts of DNA that are not used for anything. So the coding part of DNA actually carries information. Like they will give information how to make insulin, how to make glucagon, how to make enzymes, how to make this, how to do that, how to do that. So those are coding areas of DNA. Then we have non-coding areas of DNA that seem to have like no information in them. Like they seem to be there. We have these regions of DNA, they have the nucleotide bases, but they don't seem to actually give any information related to the body. So those are known as non-coding region. So the function of non-coding region we don't know, but once the messenger RNA is made, it generally has both. It has coding and non-coding DNA, because when you're making the messenger RNA, when you're doing transcription, you are basically just making a complementary basis. That is it. So when you're doing transcription, you're just making a messenger RNA using complementary basis. The entire section of DNA is basically transcribed. So it comes with both. The coding region also ends up inside the messenger RNA, and non-coding region also ends up in the messenger RNA. So what we have to do is we have to remove the non-coding region, because like I said, non-coding region does not have any information on it. So we want to make sure, for example, if you're making insulin, that the RNA has all the information related to insulin. So then the messenger RNA is modified, and the non-coding region, we will remove all of the non-coding region. So whatever region we have on the messenger RNA that does not code for anything, we will remove it. So in the messenger RNA, we only have the coding region, the one that has the information. And then that messenger RNA will then move out of the nucleus through the nuclear pore, and it will pass on to the cytoplasm. Is it a bit more clearer now?
Yes, please. Yes. Yes.
No, Aisha, I can't hear you.
Yeah, it's clear. Can you hear me now?
Yeah, I can hear you now. Yeah.
It's clear, Miss.
Okay. All right. So once this entire process is done, then you move on to your actual process of translation. So translation is going to be when you are taking this entire messenger RNA, and it is entering into the cytoplasm. And now the codon on the your triplet code on the messenger RNA has to be translated, and the corresponding amino acids have to be attached to each other to make your protein. So who actually helps in doing this? Ribosomes. Ribosomes are made up of RNA, and generally ribosomes have they have two subunits. There are two parts of it. It has a large subunit and it has a small subunit. So ribosomes generally, they're made up of RNA, and they have a large subunit and a small subunit that join together to work, that join together to actually do the process of translation. So ribosome has two subunits, large subunit and small subunit. And generally speaking, so in eukaryotes, we have 80 S ribosomes, and in prokaryotes, we have 70 S ribosomes. And ribosomes can be free or they can be attached. Free ribosomes are found within the cytoplasm. Attached ribosomes are found attached to your rough endoplasmic reticulum. So your ribosomes can either be floating around freely within the cytoplasm, or they can be attached to your rough endoplasmic reticulum. So that is why, when you have studied cell biology, one of the major functions of rough endoplasmic reticulum is protein synthesis. This is why, because they have ribosomes on their surface. So they are going to, messenger RNA is going to come. The messenger RNA can go to the rough endoplasmic reticulum, or it can go to the free ribosomes, whatever. But it will go to the ribosomes, and then the process of translation is actually going to start. So we are going to, okay, we'll stop it over here. And so we have to do the process of translation after this, and then we have to basically go over our topic of mutations. So we have to cover our mutations, and specifically in mutation, we have to cover sickle cell anemia, how the sickle cell anemia mutation actually takes place, and like details related to that. And we have to do the entire process of translation. So we will just stop it over here for today. And then we'll start the topic of translation in the next class. All right?
Okay, Miss.
Okay. All right. Thank you so much, Aisha. I hope that was all clear. No confusion related